everybody. We are slowly starting the breakfast today. Today's breakfast is, as you see, with a strong group from Discovery Life Sciences. And you are broadcasting, I would say, from Kraków, yes?
Yes.
Okay, this time from Kraków, every time from Kraków. And we have strong representation of the company today, and the subject is very, very interesting. I advise you to show up, say hello before we start. You have 1 minute to show that you are here alive. It's not your avatar. Who wants to say hello? Tomasz Kwiatkowski, if you don't know, is our new employee in Cluster. So say hello to Tomasz.
Nice to see you.
You will meet with him more often now.
Hello, it's nice to see you.
Nice to see you. Nice to see you.
You all, thank you for joining.
Hi, I'm Renata from Real Research. Nice to see you.
Nice
Hi, Renata.
to see you.
So as you can always say hello when you start thinking that's time for you to show on the screen. I will pass the voice to Tetyana, because the subject is long, and then I suppose and I expect that we'll have a discussion during and after the presentation. If there is a presentation, I don't know what is the scenario for today. But okay, so because that's your day on your breakfast, so I'm grabbing the coffee. I don't know, is this here? Yeah. And start listening.
Thank you, Kazimierz. So I think that it's a breakfast. Yeah, it's not a conference. So that's why we will try to present information that we prepared today, but at the same time to leave some time for questions, for discussions, because their theme for today is really very innovative and interesting. So first of all, thank you for joining. Most of you already remember Shawn's presentation last year, which was dedicated to DTC, dissociated tumor cells. And at that time, 1 year ago, it was one of the new products that Discovery Life Sciences presented on the global market. And probably not today, because we already presented tumoroids some time ago, not, let's say, several months ago, let's say. But for today, we are proud to say that Discovery Life Sciences globally presented to scientists new product, it's tumoroid. And I think that it's a good place and a good chance to share with you what we have for today in our portfolio. Also, this is a unique opportunity to participate in this breakfast, which we have a passion for this type of activity. And taking into account that today we have a group of our top leaders of Discovery Life Sciences in biospecimens in Kraków. So, and can you put the next slide? So, Shawn, Vice President of Laboratory Operations at Discovery Life Sciences, will present information about tumoroids. Nathan Henson, Vice President Global Biospecimen Operations, so will join us in discussion if you have any questions regarding cooperations in biospecimen procurement globally. Michelle Bauer, Vice President Global Biospecimens Project Management. So we are proud to say that we are able to manage their very customized, very difficult, very, let's say, presented and prepared with care for specific demands of our clients' projects, oncology and non-oncology. And myself, so you know me also, Managing Director of Discovery Life Science Polska and Ukraine. So,
and I think Shawn is for second time on our breakfast.
Second, yeah. Second, and his profile also available on the Life Science web. So please use this opportunity after presentation if you have any questions or you want to contact him.
Welcome, everybody. Thanks for joining today. So brief agenda for the discussion today. We'll go over an overview of Discovery Life Sciences. We'll start fairly high level and work down to specifically talk about our tissue-based solutions, focusing primarily on tumoroids. We'll have one slide on tumor model history, kind of where we started with in vitro tumor models and how they've grown over the last half century. And then we'll focus primarily on tumoroids for the most of the talk, how we develop and characterize those, and then how we've moved them now into starting to evaluate new therapies that are either in the market currently or about to come to market. So we go to the next slide. So for Discovery Life Sciences, the broad overview is we have two large key areas. One is our biospecimens and cell biology services. Those are seen in the three orange circles, and that is corresponds then to our specialty lab services, which are in the purple circles. We're going to focus mostly in the orange today, but for the purple circles, we do offer primarily clinical trial support around molecular pathology. These are things like immunohistochemistry for ongoing clinical trials, various molecular downstream assays such as sequencing or PCR, which is then translated into our genomics capabilities. These are all next generation sequencing capabilities. We then support primarily not necessarily treatment decision-based technologies, but more exploratory technologies around clinical flow cytometry and clinical proteomics. And I will touch about proteomics a little bit during this talk as we started to branch over from biospecimens into using their services. On the biospecimen side, we do have three main focus areas: ADME tox. Those are primarily hepatocyte, liver-based products where we're looking at inherent toxicity of new compounds. Cell and gene therapy division, which is focused on large-scale leukapheresis collections, which I'll talk about a little bit in a couple slides. But primarily today, we're going to focus on our biospecimens and their downstream services. Let's go to the next slide. So we are primarily based in Europe and the US, kind of broad-based geography. Again, labels are color-coded. So if we're in the orange, those are our biospecimens-based sites. Blue is our ADME tox service. So we did change the colors up a little bit on this slide. Purple is those specialty lab services. And then the one that shows the gradation is basically headquarters. So headquarters is in Huntsville, Alabama. That's where the three of us are based. And it offers both the biospecimen solutions and specialty laboratory solutions. Specifically talking about biospecimens, we do have both the east and west coast of the US, as well as multiple sites here in Europe, including Poland, Ukraine, Romania, and Bulgaria. So for each of these sites, there are some specialties. The sites in the US on the coast primarily focus on that cell and gene therapy division. So large-scale normal donor collections. The site in Seattle is where we do a lot of our hematology toxicity assays. In Huntsville and then in the European sites, we focus primarily on our disease biospecimens. That's most of what we'll talk about today. Heavily in the European sites focused on tissue-based procurement. A lot of what we do in the US is blood-based procurement. So we go to the next slide. So what this looks like then in that global clinical network is that we can see up to 20,000 tissue collections per year and up to 50,000 biofluid collections per year. And this gives us really a robust inventory of samples that we can provide. So most of that inventory is hosted in Huntsville. We do have a very expansive freezer farm that we call it, which holds all the viable biospecimens, and then the bunker or warehouse, which hosts our fixed tissues. But we also do this prospectively. So not necessarily a collection that needs to go to one of our labs and then to the repository, but direct to downstream user. And that's really what Michelle and her team manage, is making sure that all those products get to the end user as quickly and as safely as possible to be utilized. So we go to the next slide. So when we talk about biospecimens, we talk about it in basically three big buckets. The bucket on the left side of the screen, that's all of our blood-based products. So the top is those apheresis collections. So in this case, we have normal donors come in. They provide blood, but in this case, we've enriched it for specifically white blood cells. Those tend to be very valuable in the cell gene therapy market, lots, billions and billions of cells in these collections, and then we can take it into various downstream derivatives. On the normal side, but primarily on the disease side, we also provide just standard whole blood. Again, that can be shipped fresh, or we will process it to either the liquid components, so plasma serum, or double-spun plasma, which is very popular in the diagnostic market, as well as buffy coats, so just that white cell component, or viable PBMCs. On the right side of the screen are our tissue-based solutions. So in this case, we do include bone marrow. It kind of branches the blood and tissue, but as a tissue-based solution, ascites fluid primarily from our ovarian cancer patients, which there are some tumor cells present in, hepatocytes from our livers. But in the tumor world, in the upper right corner, flash frozen tissue and fixed tissue. We won't talk about that today, but then dissociated tissue and tumoroids, and that'll be the main focus of the talk. The third bucket, which is, there's always a third bucket that just is other, and that's any biospecimen that doesn't fall within blood or tissue. So swabs, urine, cerebral spinal fluid, synovial fluid, all of that goes into that third bucket. So we go to the next slide. So a brief overview. So as Tetyana mentioned, for this talk last year, I talked pretty exclusively on DTCs. So just a couple slides to level set and to bring to the forefront what those are. So we a lot of times used to provide fresh tissue to downstream users, but with such an expansive network and with users all around the globe, that's really hard to get to an end user in a time frame which the tissue can be utilized. So instead, what we do is we bring that tissue in-house to two main facilities, one in Huntsville and one in Bulgaria. It's minced, or actually, and Tetyana and I are going to discuss while I'm here, is hopefully in Ukraine very soon as well. We'll be doing this
in a month.
In about a month's time, yes. Actually, the equipment just got there this week, which is nice. So that solid tissue comes into the lab now, is minced, and over about an hour time with some enzymes and mechanical manipulation, we get single cells. And basically what we're now doing is taking what was a solid tissue, making it a single cell suspension and cryopreserving it. We essentially have a fresh tissue in a tube. And since these are viable, they can be utilized for any of the downstream assays that the fresh tissue would have been utilized for, but the scale is way bigger, whereas you might only get 1 or 2 lung tumors a month. Fresh, we have hundreds in the inventory that can be pulled immediately for use, and we do that quite frequently. We do post-thaw QC on all these samples, so we know what cells are present in them, what their viability is, their sequencing profile is. And so really, again, as we were talking about personalized medicine before we hopped on the call or on the presentation, when you're looking for very specific patients, now you can get very specific tissues from the biorepository that are viable and ready to use. Let's go to the next slide. So there's a lot of words on the slide. So basically, the crux of this slide, or the overall idea, is that these are highly characterized. So if it's not coming from the pathology reports that we receive from the sites, we are further characterizing them for things like microsatellite instability. How is the genomic instability of the tumor? Are they HPV positive, specifically in cervical and head and neck tissues? PD-L1, which is a key target of a lot of current immunotherapies for things like lung and head and neck tissues. We also do high resolution HLA typing, the estrogen and progesterone receptors, as well as HER2 on breast tissues, really trying to get as much characterization so that when cohorts are built, they're built with the knowledge of, hey, these express or do not express the key targets of my new therapy. We have expanded this, and so as we initially started this years ago, as DTC started for tumor, now that T stands for tissue, as we now have normal tissues, primarily lung, colon, ovary, but we also have autoimmune tissues as well, ulcerative colitis, Crohn's tissue, and are going to hopefully expand that more as we move forward. We do some targeted sequencing for mutations and fusions, and as with all our biospecimens, we do have the base patient data, as well as those pathology reports. And so really it's a very, not only from the specimen side, but from the data side, a very robust
and even follow-up data.
Follow-up data, which is even more crucial, actually, as we get into the tumoroid models. Yeah. Yes, having this follow-up data has been monumental, and actually some of our selections for tumoroid generation. So for these DTCs, we can put them into a number of assays. This is, again, what I focused on a lot last year. So they can be sequenced both at the bulk level, which is the heat map in the top left, but at the single cell level. So those little colored dots are essentially what a colorectal cancer tumor looks like by single cell sequencing. Flow cytometry, which is by far what we've done the most on at this point. We've done flow cytometry in about 10,000 unique tumors. The functionality of these cells, so can we take them out of the thaw and make them perform like they would in the body, which is what the bottom right is. But really the bottom left is the rest of what we're going to discuss today, which is in vitro cell culture of these. And can we use these to model in vivo responses, particularly to new therapies? And so that's where we'll spend the rest of the time talking. So as we talk about tumor models, just one slide to kind of go over the history of tumor models, which really started back in the 1950s with the first cancer cell line, HeLa cells, a cervical cancer cell line that came out of Baltimore. And that really revolutionized the ability to do in vitro testing as well as understand specifically indication characteristics at scale. And so over the last decades, last quarter century, we've really expanded the number of cancer cell line models, and even though I will talk about their limitations in the next slide, really that they are still fundamental to the development of new therapies. Once we got into the 60s and 70s, you now see the advent of mouse tumor models, again, very, very powerful, but very species specific. And by the mid 80s now, we've had enough cell lines generated that we can start developing large cohorts to do screening platforms on. Really, as we moved forward, though, into the future, into the 20s, what we noticed was the need for primary cell culture. So mouse models were great. They provided us the opportunity to do some in vivo work, but they are still mice. And so these tumoroid models, which we're going to talk about today, are really in vitro models from primary cells. And so what you'll see that the little movie on the side there, that is a lung cancer tumoroid model growing over the span of a couple days. These are physiologically relevant. So they aggregate into three-dimensional structures that mimic the inherent primary tissue. They have all of the genetics of the primary tissue. And most importantly, they can be scaled. So a lot of the earlier models that tried to do this, they either had short time frames or low cell numbers. With these, we can grow them up to massive quantities and freeze them back, which allows you to go back to the same patient multiple times, especially as new compounds or new therapies are produced, to really understand how the same patient would have responded. So you're basically just getting more and more data, but you minimize the number of variables as much as possible using these models. So we go to the next slide. So if we look at the cell models that have been around for half a century versus these new ones, immortalized cell lines, again, very powerful. They're very easy to grow. You can use them a lot for biomarker evaluation, initial drug study, but they do have the tendency to drift genetically from the parent tissue, making them very clonal. There's limited complexity there, whereas tumoroids remain as complex as the parent tissue. Fresh tissue, again, very valuable, but it's very limited in supply. And getting to that downstream user is very difficult. And primary cell culture is very, very restricted in those. Tumoroids are banked. They can be propagated long-term, which we'll show. Animal models, again, great, especially for in vivo responses, but you can get some species specific. When I was in school, our professors always used to like to say, we've cured cancer in mice hundreds of times. We still haven't cured cancer in humans. So while they can be valuable, they are not exactly translatable to humans. And so in these tumoroid models, there is no animal. It is an in vitro model, and it is three-dimensional. So 2D models, again, they tend to lay on plastic, which we are not plastic. So our tumors are not growing on plastic. These are three-dimensional. They grow in a suspension matrix, and you can grow them long-term. These have been taken out almost a year at this point, 20+ passages. We're always check genetics at every 4 to 5 passages. It always matches up back to that parent tissue. And then finally, the spheroid models, which we do use for short-term cultures. They don't exactly mimic in vivo conditions. They tend to get a little necrotic, where these models that are tumoroids do.
Shawn, does it mean that it's just these cells are replicating, but they have no chances to any kind of changes in the conditions, in the laboratory conditions, so they cannot mutate, yeah? So it's just replication.
Exactly. They do not mutate more. We've seen some slight changes, but they're largely percentages of mutations. It's not really the mutation itself.
Yeah, thank you.
So the models that have been developed so far, we have about 15, primarily focused in colorectal and endometrial cancer. Those are the two cancers that were most amenable to the system. We've also developed some lung and breast cancer models. And our president has told us that we need 100 of these by the end of next year, which is stressing the lab out just a little bit. So you'll see this list expand pretty quickly. But one of the keys that we've done here is that we don't just have the tumoroid itself, right? So this is a pure tumor fraction. Tumors themselves contain immune cells. They contain support system cells like fibroblasts. So we started to match a lot of these with a lot of those support systems. So we've generated cancer-associated fibroblasts from 3 of these lines, and going forward, we're going to try to get that from as many lines as possible. We have the parental dissociated tissue cells still available for a couple of these. And for most, correct me if not all, of our tumor collections now, we are getting blood at the same time. And so this, you'll start to see a lot of these have PBMCs as well. So now you can really recapitulate the entire tumor microenvironment. And we'll show how we're trying to do that in vivo or in vitro near the end of the talk. But if you hit the next slide, really what we're giving is not only the tumoroid itself, but really a very expansive data package. So we still get those patient demographics, the pathology report that we have for every sample that we collect, but now we're getting the biomarker status, if that was done by the hospital that we partnered with, or if we do it internally. So all of these are going through MSI status internally as well. Follow-up data, like Tetyana mentioned. So one of the next lung models we're looking at is a lung tissue where we actually had treatment data against Keytruda, against a PD-L1 therapy, where we know that it became metastatic. So now we can actually test in vitro if we get the same results. We're doing some very deep sequencing. So we're sequencing the entire exome from these patients, which gives us relevant oncogene mutations. So in this case, one of our colorectal cancer tumoroids actually has a KRAS mutation, a known oncogenic KRAS mutation. We're going to start testing that in vitro against some KRAS drugs, as well as tumor mutation burden, a way to know if these would be immunogenic tumors, as well as RNA sequencing. So what genes are expressed and then what genes have fusions. Again, very important for oncogene-driven mutations. And then coming soon, I say coming soon in that we actually have the data. We just haven't figured out a way to package it yet, which is mass spectroscopy. We are doing full global protein analysis on these tumoroids, as well as drug response studies. And so you'll see a little bit of the infancy of both of those, but eventually that's going to be part of what we call the data package that is delivered with these, which is this massive Excel spreadsheet, which has all of this data to sift through.
In which way community of people, organizations, companies or universities can have access to that? It is your service, or they can have, I don't know, buy the models and use by themselves somehow?
Yeah, so these models are readily available to everybody. Actually, there was and actually for universities specifically, they have become very attractive. I had a conversation with a professor in Italy actually last week, which reached out and he said, hey, I've got tumor models that I've made from my university, but due to the way that the patients were consented, he has only limited ability to use them. Whereas our models, because we've taken care of all of that informed consent on our side, they really like being able to use these because they've got much more ability to use them for any downstream assay. So a lot of universities have started to reach out to us to start using these as well.
So in general, it's a product which is available from Discovery Life Sciences on request. Definitely with some extension of their localizations and type of cancer, which models are ready for today.
Yep.
Okay.
So another question, because I represent a contract research organization, a company for profit. Are those licensed to be used for profit?
Yes. And so there are some nuances that would happen during the contracting, Nathan, correct me if I'm wrong on that side, for a CRO to use. But yes, these are readily available for CROs. You would just have to contact us. We would work on all that on our end to get those into your hands so you could use them for that type of research as well, for profit.
So that's your out licensing. Now, the tumoroid or organoid space, so heavily patented by Hans Clevers and Hub Organoids, is the IP clean here?
Yes, so that is an amazing question. So when these were developed, yes, they were developed to not infringe on the hub technology for organoids. So we are completely separate from them. None of what we use is impacted by the patents that are currently held for those organoid models. So luckily we are clear from those.
Okay, because that's crucial for us, right?
Yeah, we tried to do the organoid models, Nathan, for a couple of years, right? We found them, and actually it's a good segue to the next slide, difficult to get scaled properly. And so in the generation of these tumoroid models, that's one thing that we focused on, is the ability to scale. So yes, yeah, this is great. So how these systems work and what makes them really easy for scaling is that we have that dissociated tissue. We initially embed it in an extracellular matrix, similar to what the hub organoid technology uses. So these are little gel-like domes where the cells are embedded within, and it really allows them to aggregate within that matrix and start to expand. But after early passage expansion, we actually moved to a suspension-based model. And so this still has a little bit of that extracellular matrix in it, but what these grow like is more like a non-adherent cell, like a leukemia cell line. So they look like balloons of cells, as you will see as we go through the presentation, which allows you to scale. So when we tried to do embedded models, we could only get a couple million cells. With the suspension models, we get hundreds of millions of cells. And so what we do then is scale up to that large bank and then freeze them back. And so we're doing all the genetics on that frozen bank, and we know that it matches the parent tissue. But what we've also done is kind of a little bit of a future proofing in that the embedded culture, the suspension culture, we have those banked back at very, very low passages, such that if our cryopreserved bank depletes, which it eventually will, we can go back to low passage, expand again, check the genetics, and that's what we're doing through each. So essentially, this gives us an almost infinite supply of these cells to continually expand out. Again, but every time we do that, we are going back to those genetics to make sure that they match the parent tissue. And so this is an example of that genetic integrity. So what we're looking at here are the varying allele frequencies of the dissociated tissue, that embedded bank, that suspension bank, and then what we call the working bank or the cryopreserved bank. So across the top here, that is the embedded bank, the suspension bank, and the working bank to the parent tissue. And if we look at our squared values here, we have about 0.88, 0.87, which is really, really high correlation. Like this, from the fact that we've gotten all the way through 20 plus passages and we're still at that high level of integrity is very important. But then as we get to the models themselves, the embedded bank in the middle compared to its suspension and its working cohort, 0.99 R squared values, and it's about the same for the suspension of the working. So we're maintaining all those inherent mutations through all of these passages that we've done. And if we go to the next slide, which was really crucial for us, was the next slide, which is these are two colorectal cancer models. And these are the two that we'll use for the rest of the talk, which is 3309 and 1104. So I'll only use the last four digits for those. But if we looked at the VAF or the allele frequencies from the DTC across both, the R squared value is 0.4. So basically no correlation. And when we get to the tumoroid, it's 0.11. So, I mean, basically showing that while it's maintained within a model, across models, they are entirely genetically different. Which again, as we move to personalized medicine, is crucial, right? We want to make sure we maintain those genetics and that we can go back and then test for specific mutations. So the next step that we took, that's our whole exome data. So we've now got all this RNA data as well. So this is a heat map of those two models. We used the 3309 model basically as the benchmark. And so we looked at all of the genes that are expressed. So this had to show some RNA expression. We set basically our transcripts per million at 1, which is very low expression, but indicative of some expression. Of those genes that were expressed, 5,000 of them had associated protein found by mass spec, which is insane, actually, that we had that much correlation. But what's crucial then as we move to the next model, the 1104 model, is that it did not match, right? So the RNA is largely correlative, but there's some gaps where the protein is completely unique. And we actually confirm that using flow cytometry. So this is looking at two very common drug targets, HER2 and ROR1. HER2 was expressed in both models at the RNA and protein level by flow cytometry, using the blue histograms, which are the specific antibodies for those, show clear expression against the isotype control in red, whereas ROR1 was only expressed in the 3309 model, so quite clear expression by the blue histogram, but the 1104 model did not express it, so it completely overlaid the isotype. So this gives us a gigantic repository of targets to which to look at drug expression or drug target expression, biomarker expression, and be able to then correlate it to other technologies. So that's all about being able to profile those samples. What really the crucial nature of these samples is to be able to test drugs. And so this is basically showing very broadly the different types of cancer therapies. And they're basically this bucketed in two buckets into non-immunotherapies and immunotherapies. The way I'm going to look at this is cell intrinsic and cell extrinsic. So cell intrinsic are those therapies that are trying to affect the tumoroids or the tumor cells themselves. So things like, well, surgery, which is a resection technology, but radiation, chemotherapy, the idea that we're trying to target the fact that tumors are proliferating more or should be proliferating more than the rest of the cells in the body. Or the targeted therapies there, small molecule inhibitors, antibody or antibody conjugates, where we're trying to take a drug and target it to a specific tumor cell. The cell extrinsic ones, or the immunotherapies, as are shown on this diagram, are those that are going to require another cell type. And so a lot of times these are bispecific antibodies, something that's going to redirect a T cell or an NK cell to a tumor cell to kill it, or adoptive therapies, CAR T cells. So taking cells from the patient, manipulating them to express against the tumor, and then re-injecting back into the patient. So I think I might have an animation here. So if we hit the next button, again, cell intrinsic on the left, cell extrinsic on the right. So for cell intrinsic, we set up a very simple system. So this is using a reagent from Promega called Cell Titer Glow 3D. It is a cell viability assay. So basically, it's measuring the number or the robustness of the viability of the cells in culture. So what we've done is taken our two tumoroid lines, 3309, 1104, place them in culture for 7 days with various compounds. So two more chemotherapy-based compounds, 5-fluorouracil, etoposide, and then a small molecule inhibitor against WEE1. And so in the red, you're going to see the response of the 1104 line, and in the blue is the 3309 line. And so x-axis is various drug concentrations. Essentially, look at the y-axis is percent viability. So both cell lines in the presence of 5-fluorouracil, it killed them very robustly. The etoposide, it didn't really have much response on either line. But if we look at the WEE1 inhibitor, very targeted death of the 3309 line, not of the 1104. So we can already see, just with two lines, differential responses to small molecule inhibitors. And then if we go to the next slide, we actually have started to work on antibody drug conjugates. So these are two different antibodies against HER2. They differ in their affinity for HER2 itself. So the compound shown on the left is actually more specific and has a higher affinity for HER2 than those on the right. These also had different payloads, so different drugs that were tagged with them. And so again, 7-day culture, but we can see that the 1104 line, even though it does express HER2, did not have much response to either antibody drug conjugate. The 3309 line did, but only to one, the more high affinity antibody, but not the low affinity antibodies. So I really like to show these examples because this is only 2 lines, only 6 drugs, and we can already start to see some differences. And so this is one of the next iterations of tumoroids. We have about 10 to 12 compounds that we're going to be testing every tumoroid line against. It's split mostly between chemotherapy-based agents, small molecule inhibitors. And so that'll become, again, another data point that we will have for folks to start looking at tumoroids to start understanding which ones might fit their research. So this is the system that we're probably the most excited about, but the one that has, quite frankly, been the hardest to kind of get into a graph or something that's easy to visualize. So this is using an IncuSite S3 system. So what this system is actually a fluorescent microscope that is hosted inside a CO2 incubator. And every whatever time course we set it at, so we set it every 12 hours, though we've done it every 4 hours as well, it will go through and scan every well of a 96 well plate. It takes about 4 images per well. And basically over time, you can graph either the growth of cells, the death of cells. There's several different metrics that it can visualize. And so what we've been doing is adding our tumoroids in, which we can see grow, and then adding different cell types in to see what response they have. So the one that we're showing on the left is basically a regularly growing colorectal model. And it's a little hard to see here, but most of the tumoroids, they're clear. You can look in, you can see inside them, they're nice and round. We've added in NK cells generated from a normal donor from our network. Now the tumoroids are more black. They don't have that nice morphology anymore, which really suggests that we've got some sort of NK cell-based killing of these tumoroids. And so we're now adding back any of 6 different systems into these models. And so we've broken them up into 2. So autologous, those that came from the same patient. So again, we're generating as many cancer-associated fibroblasts as we can. So that's the bright field image that you see there. That's a calf line. These look by morphology, by protein expression like fibroblasts. We start to add those back into the system. We have the tumor-infiltrating lymphocytes, or the TILs from the parent tissue as well, that we're adding back in, or PBMCs from the same patient. And then we have the allogeneic bucket. So these are from different patients. We're using NK cells and gamma delta T cells. The advantages of both of these immune cell models is that they are not restricted by the inherent HLA of the parent tumor. And so we can get some nice responses without having to worry about any mismatch. But we are fully conscious that there are some therapies that will not be predicated on NK cells or gamma delta T cells. And so what we've also started to do is our entire normal donor pool has been HLA sequenced. And so every time we have a new model come out, we HLA sequence it. And then, and I don't envy these folks, they will go through and match HLA matching from our tumoroid to any of the thousands of donors in our normal donor network. And I can't believe this happened, but we've actually had matches. And so when we have matches, we bring those donors in, and we have now frozen banks of normal donors. So really, the conclusions and where we're going here is this network that we've made is really crucial, this global network for advances in novel therapies. And it's because we can get robust clinical data, including that follow-up data that Tetyana mentioned, where we know how the donors responded to various treatments, and we now can test in vitro and see if we get the same response and then how other therapies would have worked, and we get those matched sample types. And it's really crucial that we have this in multiple locations across the globe. These tumors that we developed are currently the most physiologically relevant primary tumor model out there. They've not been manipulated. We've introduced no immortalization. They intrinsically are as native to the parent tissue as possible, but we've added that high characterization. What are the inherent mutations or variants of that parent tumor? What genes or what proteins are they expressing, and how are they responding to drug responses? And then there is a ton of focus in immunotherapies right now. And it really, it's fun. I'm an immunologist by training, so it's fun to watch all of this cancer immunology come out. But really, because of that, these tumoroid models, which are pure tumor models, need to have a co-culture system to really understand how those work. And that's where that IncuSite system is going to come in. How can we redirect T cells to kill these tumors in the presence of bispecific antibodies, or manipulate and generate CAR T cell models to affect these tumoroid models? And that's a heavy focus of the lab through the end of this year and next year, in addition to growing hundreds more of these models. So that's basically where we are with tumoroid models. I could give the same talk in a year and it will be vastly different, because we're going to have way more models and a lot more data on how they function. But we'll take any questions on tumoroids, or really anything that we do at Discovery. That's why I like having these folks with me. I'm very focused in the lab. They're very focused on things like the clinic or getting samples to downstream users. So we'll take any questions, open for discussion on anything that we're doing, tumoroids or otherwise. Thank you.
Thank you.
Thank you. Very interesting and very detailed presentation. Actually, for me, it's very difficult to phrase the questions regarding the details of your presentation because I'm not specialist. There are specialists on the in the room who may have questions. So I invite you to ask. I have different type of question, I would say, and forgive me if this is something kind of out of ignorance in the field. But I have an impression that we are talking still about the research which leads us to the development of the treatments to cure the cancer. Is this also useful in development of diagnostics tool or maybe preventive tools?
Yeah, so it's a great question. And that's actually a big area where we work as well as in diagnostics. And so the advantage of these models is because we have so many matched sample types, we can see responses and then go back and see, hey, was there something indicative in the plasma? We do a lot of liquid biopsy work because there's something indicative of the PBMCs or that parent tissue that we can now use diagnostically to predict, right? Again, predictive medicine here. Predict, okay, this tumoroid had a great response to this therapy. We have that follow-up data. We show the patient had a great response to this therapy. Now, what can we look in the blood that would have indicated this and use it diagnostically? I don't know if you have any other thoughts.
No, let's say knowing, even with the personalized therapy and or testing with their potential drug treatment. So if we can make a model knowing how their tumor cell will react on the treatment, so this model give us opportunity to go from the end to the beginning. So what kind of diagnostic tests results are specific for these tumor cells? So so probably if somebody will discuss such opportunities for diagnostics for potential treatment prognosing, and even probably 20 or 30 years ago, it will be like magic, pure magic, So any patient can survive if you will try on him 10, 11, 12 regimes of chemotherapy. Any patient can survive if you will try to get all possible tests and after that compare that with their reaction on the therapy regimen, But if you can build a model of the successful treatment, this theoretical, let's say in vitro, successful treatments, a successful reaction on their some molecules at the end, you can backforth to understand, try to understand what kind of characteristics for these cells at the beginning. So this is like you can use this model in both directions, coming to the future and returning to the back. So in this case, yes, this can be used also. Probably in a, because I'm not also immunology, that's why I try to translate it on the, let's say, simple language.
Yes.
But it sounds like challenge for the community. What you can propose having access to this type of tools. I was asking this question because just yesterday we did hear the very fantastic presentation here in Gothenburg about diagnostics in cardiovascular type of cardiovascular diseases, and that was it was the presentation delivered by startup, and they said actually with our technology people will not die because of heart attack. That's something which can eliminate because of the detection, early diagnosis and detection, and accurate treatment that will was eliminate the cause of heart attacks. So if everybody could be actually with the danger of heart attack of this type of disease will be treated, diagnosed, this can be avoided. And of course, there is a kind of holy grail regarding the cancer, in which way we can prevent being ill in the cancer. So this tool, I believe, I imagine, it's not the kind of believing, it's a kind, I imagine, could be used in that direction. But that's my ignorant questions. Please, if anybody from audience would like to ask questions, please go. Don't hesitate to engage in dialogue. Or it still becomes it seems to be a very difficult subject for us. Yet another question which I have is in which way we can support a community, especially those research community, especially on universities, in having access to that tools. This is only the question of buying the service from you, buying the samples, or it can be organized in different model that this access will be granted. It will be for them easier to use these tools in their research. Can we provoke research in that direction because there is a tool? That's the question also.
So in this case, how we would not necessarily just provide the tumoroid, but provide expertise in the tumoroids?
What do you mean, Kazimierz?
No. I mean, in which way we can support the direct question is in which way we can support the community in having access to the tool. I'm asking from the perspective of how to say infrastructure. Let's say, for example, bioincubators, accelerators are very often equipped with so-called core facilities. The core facility is something that people have access. They cannot. They are, especially at the early stage of the development, they cannot. Organizations, startups cannot afford to have their own. But it's enough to have access to something, to the facility which is useful in the development. Yeah.
Yeah. Okay. Yeah. So the other thing that we offer is and that we do quite a bit is, we're not just growing these, we're testing them. So at any one time we have 5 to 10 of these lines out. And so for startups, we actually get a lot of these where they'll say, hey, we're a virtual, or we haven't built our lab out yet. Can we just work with you guys and send stuff and we test? And that's 1% of them we do. So in this case, yes, you don't even have to bring the tumoroid in-house. Just here are my compounds. Can you give me some good readouts? Or, hey, I'm looking for this. We had a client recently who reached out and just said, hey, I just need to know the expression of this marker. Like, can you just let me know if the tumoroids express it? Like, yeah, we've got the data. We can search through it for you. So yeah, that's something we provide as well. It's not even just the physical vial, but the service to test it, to help build what it looks like. We did letters of support for a group recently who was trying to get some funding and said, hey, if it would help them out, if they get the funding on how to develop this next model. So
technically speaking, it can be organized again under the brand of, let's say, core facility, but it will not be the physical, but the digital facility, I would say. So access to the service, to have access, or to get access, or to be serviced by you because of the huge data sets you have, which can be useful for some kind of developments.
Exactly. Yeah.
And I'm talking as an ignorant in that field, so you have to pick up from that what is useful or not.
Yeah. Well, I think the other thing is, we talked about all the data that we've got, especially sequencing wise on the tumoroids, but we've got a huge endeavor right now to do that just on our biospecimens in general, right? I mean, how many have we sequenced at this point?
3,500?
Yes, 3,500 samples that have undergone sequencing, and we're doing a big initiative next year to add a couple thousand to that. And so that data alone is incredibly valuable. And so we do periodically have requests like, hey, can I see what that data would look like?
Also, we can mention that we already have such clients, and we're, let's say, discussing different opportunities with them, which is really interesting and appears not so long ago. So they really want only their digital data on everything we did. Yeah. So they are not interested really in physical biospecimens, or so they need only their digital images if we speak about tissue, and digital are already, let's say, this mass data which can be analyzed. And we believe that amount of such requests and they are also servicing, like we are servicing our clients with the well annotated biospecimens. They are servicing their clients with their how to say, well processed digital data. Yeah, so like a next step. And in any case, this is something that will be more and more often requested. We understand this absolutely clearly. And it will be not something in 2-3 years. No, it's happening just now. So even during 1 year of our different negotiations and cooperation with such companies, we see the dynamics of their requests and the progress they are making. So this is really something that will change the way how the data is arriving to the researchers.
So, any questions from audience? Sorry, but I still have some ideas to explore, at least. Talking about this diagnostic approach, the approach from the diagnostic perspective, would it be possible to useful for a hospital having access, some kind of the immediate access to some kind of service that would help them to quickly diagnose or confirm diagnosis or whatever, I would say online with patients in the hospital?
It's a good question. I think what Tetyana was talking about is probably the closest that we're coming now, which is this AI, basically AI-driven analysis of whole slide images.
That's direction which I wanted to provoke, because AI may help
much closer to you, Kazimierz.
Yeah. I mean, we're working with some AI companies right now, basically on these data sets that we have. Nathan probably can speak more to that, how that's going. But yeah, with whole slide images, providing we've provided a lot of whole slide images to AI companies to start modeling. Could they predict responses? Could they predict biomarkers? We actually did one for MSI status where they had predicted microsatellite instability or genomic instability by the H&E through an AI algorithm, and then we confirmed by a conventional wet bench process.
I believe that what you are asking, so this is something that will more or less routine and much more sooner that we can imagine. The problem that the methodology and what Shawn presented today need to be scaled to make this attractive and available for hospitals on the daily basis. It needs to be a kind of the routine, So I cannot imagine that for today, even smaller companies or laboratories are able to provide such detailed annotations, because it's very expensive. You need a lot of equipment, and patient cannot wait for 2-3 months. I don't know when you will have a, let's say, waiting line to receive all these samples annotated. But without this preliminary stage, what we're doing now, and now this is available for scientists, it's available for research facilities, but the next step will be what you asked at the beginning, probably proposing some kinds of the tests, AI mechanism, whatever, because you need to have a big amount of these studies statistically to be reasonable, for other steps. And yes, everything that happening now probably have their mission and vision to make it available on the hospital level. So you can take tissue tumor cells from patient, you can replicate those cells how many times you need, you can test all molecules on that and define the really personalized treatment for this patient. Yeah. But in any case, nobody will be, so it's impossible to imagine that hospitals will be equipped with a so big amount of equipment. Yeah. But if basing on what we're doing now and what we're providing to scientists, they will be able to develop an AI model. Yeah. For the specific tumor cells, specific tumor types. Yes, it will be cheaper. And this cheaper means available for hospital-based usage. Probably it's, like all scientists are dreamers. Yeah, and this is the most exciting part to be scientists. But I believe that this is not something that will happen in 10 or 20 years. It's something that will happen probably in 3-5 years. Looks like that.
Would it help if the population was totally sequenced?
Yeah, definitely.
And I think that's where we're going, and that's where I like these tumoroid models, is by sequencing everything, mass spec everything, drug response everything, then you can tell the AI, here's my data, and have it sift through what was an insane amount of data to find correlation.
Still some place for mistake, but the probability of this mistake will be minimized.
Yep.
Okay, so Tetyana, let's talk next week. I have some idea for you. Okay, so we are any questions? Any other questions? Please come up. If not, I will do some. Thank you very much for this presentation, and also for answering my questions, providing they were not stupid. So that was nice. And some announcements. I remind you that it's LifeScience Open Space is approaching 27-28 of November. The stage is already open for presentations. I urge you to. I'm talking to cluster members especially, but to all present here to propose your presentations. That's the event is to showcase how we can. What we are offering to the world, the rest of the world, in which way you want to, in which way there are opportunities to collaborate with us. So it's kind of a good opportunity. I've been talking here in Gothenburg with many people who really don't know. What we are doing, what's going on, where are the opportunities. So this is the chance, and so please do that. But sooner, next week, we have the breakfast, cluster breakfast like today, and this breakfast is live this time. It will be with company Virumet, cluster member Virumet, and it will be something about simulation hubs. So something you would like to touch, not only to listen and talk about. And that's why it will be live. We meet at 9 at Dynia restaurant, and I forget the street. That's Krupnicza 20. We meet at 9, but just after the breakfast, there will be also EIT Health Morning Talk meeting in which we will be discussing the hospitals of the future. So that's the subjects are still present with us, and they are provoking to connect the dots of different activities. Please come. I invite you to come to this breakfast. To these two breakfasts, I would say. The breakfast is free in that time. Coffee, cake, and something else will be served. But a great opportunity to engage in discussion live, and also a great opportunity to experience some of the developments from cluster members. So if you didn't do that yet, please register. And then also, if you didn't do that, register to the LifeScience Open Space. Some surprises during the LifeScience Open Space will be as well. Okay, that's all from our side. We are exactly at the point. This is the 10th hour of today's day.
Yeah. Thank you so much.
Thank you.
Thank you very much. See you next week.
See you. Bye.